Many cultures burn the dung of livestock for both ritual and practical purposes, including cooking, heating, and waste disposal. In archaeology, burnt livestock dung can be found in numerous contexts such as campsites and hearths. In southern Africa, archaeologists have identified multiple burnt and vitrified dung features that begin to appear in the landscape during the first millennia CE. The reason these contexts are burnt is not entirely known. However, ethnographic research suggests that it could relate to ritual practices or the health of livestock, that is, to prevent the spread of disease. The extent to which these burnt and vitrified dung contexts preserve organic materials, such as DNA, hormones, and botanical remains, is still not entirely understood. Recent research into the presence of hormones in mottled cattle dung suggests that temperature can greatly affect preservation. And in archaeological contexts, pollen and botanical remains have been previously detected in the dung at Iron Age kraal sites. More is known about the general relationship between organic remains and preservation based on temperature, but not in the specific context of burnt dung. Based on these factors, the goal of our project is to improve sampling strategies for organic materials at Iron Age kraal sites by studying the chemical changes that cattle dung undergoes as temperature increases. The objective is to develop a means to target specific sections of burnt dung features based on the temperature that the dung reached. In order to accomplish this, we intend to burn modern samples of cow dung under controlled conditions, and then analyze these samples using Fourier-Transform Infrared Spectroscopy. If successful, our data could be used to create a temperature gradient, which could aid in identifying samples most likely to preserve certain organic materials. For this study, we collected samples from a farm in Durham, Connecticut, owned by Yale professor James Scott. These samples were produced by two Scottish Highland long-hair cows. Multiple samples were taken in order to avoid the risk of anomalous readings. All samples were weighed before the heating process. We chose a consistent mass of 5 grams for each. Once prepared, the samples were placed in a thermoscientific brand muffle furnace at intervals of 100 degrees Celsius, ranging from 100 to 1100 degrees. The 1100 degree threshold was chosen because this is the point at which dung has been known to vitrify. Each sample was burned for 10 minutes. In addition, a second burn of 20 minutes was conducted at the vitrification point of 1100 degrees. We then analyzed the samples via Fourier-Transform Infrared Spectroscopy, or FTIR. The analysis was undertaken at the Clean Laboratory in the basement of the Department of Anthropology at Yale University, using a thermo-scientific Nicolette IS5 FTIR ATR. We chose FTIR because it has been utilized in other research to determine what temperature bone and ceramic remains have reached. FTIR analyzes material composition by shining a series of multi-frequency light beams at an object and measuring which wavelengths are absorbed. FTIR has an advantage over other widely employed forms of spectrographic analysis in archaeology, such as X-ray fluorescence, which can only analyze the elemental makeup of a sample, whereas FTIR is capable of detecting complex molecules. Before each sample was run, a background measurement was taken and an automatic baseline correction was performed on each of the spectra. Cattle dung is an organic composite material that is comprised primarily of phosphorus, nitrogen, carbon, hydrogen, oxygen, and faunal bodily and flora compounds that include urea, cellulose, and lignin. Because of the non-uniform makeup of dung, our FTIR results proved somewhat inconclusive. Between 100 degrees Celsius and 500 degrees Celsius, the FTIR results show a change in absorbance between 3200 and 3650 wave number. This portion of the wave number scale corresponds with OH, or oxygen-hydrogen stretch. This change can be best explained as water loss through evaporation. If you look at the 100 to 500 degrees Celsius FTIR graph, at the wave number peaks between 1800 and 1000, there is a fairly clear relationship between increased temperature and decline in absorbance. These wave numbers correspond to several organic compound groups that include molecules such as anhydrides between 1050 and 1040, and cyclic alkenes between 1650 and 1566. The breakdown of these wave numbers is likely due to the combustion and destruction of organic plant matter remains and cattle bodily waste. Past 500 degrees Celsius, the FTIR results become less clear. As you can see from the graph, the portion of the wave number spectrum associated with organic combustion does not continue the same relationship between the temperature and decreased absorbance. For instance, at around 1030 wave number associated with anhydrides, the absorbance jumps all over the place between temperatures. From the 900 degrees Celsius sample, the 1020.55 wave number has an absorbance of 0.415, and the 800 degrees Celsius sample has an absorbance of 0.337, and the 1100 degrees Celsius sample has an absorbance of 0.098. Past 500 degrees, our study did not find a clear relationship between wave number, absorbance, and temperature. While more experimental work is needed to confirm this fact, these results indicate that FTIR is primarily beneficial for discerning between two temperature categories: a 0 to 300 degrees Celsius dung group that shows minimal water loss, and a 400 to 500 degrees Celsius category which displays clear signs of water loss through water evaporation and the combustion of organic floral and faunal components in the dung. While our results are somewhat disappointing, given that many of the kraal features throughout southern Africa have vitrified dung, which reach a temperature of over 1000 degrees, it is likely that the sections of the kraals below 500 degrees Celsius would preserve the most organic remains. In the future, our team hopes to run more samples from multiple individuals located at different farms. Because dung is an organic composite material, there is compositional variability between individuals and regions based on the diet of different cattle. Diverse sampling would allow us to create a database, which could be compared to archaeological dung samples from all over the world.